Okay, 5.2 phytohormone. What regulate responses in plants? Now, in human beings, still remember back in Form 4, we have nervous system and also endocrine system to regulate our daily activities. But for plants, they don't have nervous system and endocrine system. That's why the response of plants towards the environment can only be seen clearly after a period of time. They don't have nervous system, endocrine system. So phytohormones, this is the terms known as the plant hormones, which are the chemical substances that will stimulate and coordinate responses in plants at low concentrations. They are synthesized at certain plant organ, then they are transported to a target organ, and it is through this transport structure known as the phloem, the one that transports organic substances. Okay, so here the functions of phytohormones, we will be looking at the types. Okay, first and foremost, the famous one, the important ones, this is the auxin. Okay, so let's recall on the system in a plant, we have the shoot system and also the root system. So shoot system here is the one above the ground. Okay, we have the stem and also the leaves, the fruits and the flowers. They are the one that produces sugars by photosynthesis, carry out reproduction, those are fruits and also flowers, they are the reproductive organs. And also we have the root system, which is in the ground, that anchors the plant, that penetrates the soil and also absorbs water and minerals. Remember that the root is the main organ in charge of these. Okay, also some of it serve as the food storage. So let's look at the importance or the functions of auxin. Okay, we have here functional in phototropism and geotropism, very essential. These two ini dua, phototropism dan geotropism. Okay, so here I put it 1, 2, 3, 4 to help you to remember that you have 4 words of which starts with stimulates. Okay, so the first one is development of the apical dominance in shoots and the roots. Apical here refers to apex di bahagian hujung atas dan hujung bawah. Apical dominance punya development. Okay, number 2 is that growth and elongation in the root and also the shoot cells. So again, we are referring to these two, the parts ataupun the organs of the plants. Also, you have development of adventitious roots at the end of stem cutting. Contohnya batang tumbuhan yang kita tanam lalui keratan batang, you cut certain parts of those suitable or the selected stem. Okay, lepas tu kamu cucuk dekat tanah. Okay, that is stem cutting punya teknik. Lepas tu dia akan ada tumbuh adventitious roots. So what is actually adventitious roots? It refers to those plant roots that form from any non-root tissues. Okay, non-root tissues. Contohnya dia berasal daripada batang. Okay, kemudian bila kamu tanam di tanah, dan dekat situ akan ada akar-akar. So those akar, those are the one that from the non-root tissue, daripada asalnya daripada batang. So we call those adventitious roots. Okay, next one, the fourth is to stimulate cell division. Very essential. Okay, pembahagian cell in the cambium. You still remember the cambium during secondary growth. Okay, the meristematic tissue. Okay, so inhibits, ada dua inhibits dekat sini. So inhibits abscission of fruits and also young leaves. Abscission. Abscission means shedding ataupun keguguran various parts of an organism. So such as plant dropping a leaf ataupun fruit, the flower or the seed. Okay, these parts being shed off from the plant. So auxin inhibits ataupun stops ataupun preventing fruits and also young leaves from shedding. Okay, also it inhibits growth on lateral buds. Lateral tu di bahagian tepi. So this tunas bud is a bud that develops in the axial between a petiole and a stem. So masih ingat petiole dan juga stem. Kalau ada something bud out from here, that is the lateral bud. Axin is going to inhibit the growth of the lateral buds. Okey, kita lihat ini sangat penting. Effects of auxin on growth responses. So, plant respond to stimulus in the form of growth. Okay, kita cakap plants itu dia bergerak balas towards stimulus ataupun changes. Okay, rangsangan, kenapa dia bergerak balas? Actually, it is actually in the form of growth lah. Semasa dia membesar, so it's showing some of these response. Okay, so this response is regulated by this auxin. So now we are going to look at the effects of auxin on growth responses. Dengan melihat kepada eksperimen ini, the effects of auxin on coleoptile growth at the shoot tip. Okay, coleoptile ini ujung atas itu, paling atas, the tip. Kita panggil dia the coleoptile. So you have experiment 1, 2, and 3. Okay, the tip is cut off. The tip is cut off, all 3. Okay, so the first one is that the tip of the coleoptile is removed above the elongation zone. Masih ingat kita ada 3 zone. Zone of cell division, elongation, and also differentiation. Alright, so here di bahagian elongation zone, it is being cut. So after 2 hours, no growth is observed. Okay, after 2 hours, you put the tip back and you see the coleoptile grows upwards pula. So something in the coleoptile that is responsible for the growth of this shoot. Okay, experiment 2, where you have the tip being cut off. But then this tip is that you are putting it on top of an agar block. Agar macam ni diletakkan di atas. After 2 hours, the agar block is placed off center on the coleoptile. Sorry, this is it. Sorry, this one is referring to experiment 3. Okay, now this is it. It says the agar block is placed on the cut coleoptile. Okay, and that the coleoptile grows upwards. Maksudnya kamu sudah place the tip of the coleoptile on top of the agar. After 2 hours, you are going to put this agar on the cut coleoptile. Tapi you remove this the tip. So what can be seen? The coleoptile grows upwards. Okay, so something which is in the agar over here is responsible for the elongation of the shoot. Okay, experiment 3 is that the tip of the coleoptile is placed on top of the agar also. After 2 hours, the agar block is placed off center. Maksudnya jangan letak macam ini, but that you put it off center. Jangan letak di center. That means off center lah. Letak di satu belah. Okay, so observation is that, okay, you leave it, and that you get to see observation afterwards. You see the coleoptile growth is curved. Dia condong ke satu arah. Okay, bermaksud this part has got elongation. Okay, manakala yang tidak ditutup dengan agar, okay, there's no elongation. So something which is in the agar, okay, has diffused into this region, which is responsible for elongation of this part, which resulting in this coleoptile growth to be curved ataupun its pointing to one side. Okay, so let's look at the explanation. What inference can be made? This experiment ini is showing to us that auxin which has been synthesized, okay, dia dihasilkan at the tip of the collectile. Dekat sini hujung ini at the tip ada auxin. Okay, so this auxin is responsible in stimulating cell elongation. Okay, that is point number 1. Dia punya dapatan. Okay, number 2 is that auxin distribution influences the direction of the shoot growth. Okay, distribution of auxin. Kalau uniform distribution, okay, it will cause the collectile to grow upwards. Kalau semua bahagian ini rata, dia mendapat auxin. All this surface over here, okay, it has enough amount of auxin. That means uniform distribution. Dia akan tumbuh ke atas secara menegak. So the area which receives more auxin will grow faster and cause the collectiles to grow to the side that receive less or none of the auxin at all. So this refers to the third experiment. Sebab bahagian sini blok agar ni ada auxin, so it diffuses downwards, and therefore bahagian ini memanjang dengan lebih cepat, receives more auxin, grow faster. Therefore dia akan curve ke arah side arah sini yang menerima less auxin ataupun none of the auxin at all. So tropism is closely related to auxin distribution. Okay, tropism. Tropism, that means the directional movement. Okay, directional movement. It is closely related to this hormone known as the auxin. So external stimuli like the light and the gravity, okay, because kita sudah tengok. Sorry, over here. Now this is the one functional in phototropism dan juga geo. That means cahaya dan juga gravity. So that's why it says external stimuli like light and gravity greatly. They are influenced by the auxin distribution in plants. So auxin produces different effects to the cell in the shoots and also in the roots. Okay, remember just now I showed you phototropism and geotropism. So high auxin concentration stimulates cell elongation in the shoots. Macam inilah. Ada banyak auxin, so cell elongation stimulated. Okay, itu memang dia punya fungsi. But then, in the roots, auxin will inhibit cell elongation. So dia adalah bertentangan. Okay, so stimulation, it causes stimulation. You see auxin concentration semakin bertambah. Right, so what happened? It stimulates cell elongation. Roots. Okay, kalau dia makin bertambah dekat sini, so you get to see it is going to inhibit cell elongation. So ada dua hujung di sinilah at the shoot. It serves to stimulate, and dalam bila keadaan root over here, we have two extreme end. One serves to stimulate, the other one serves to inhibit. Shoot, root. Okay, so let's look at these roles of auxin in plant responses.